Hello, my name is Catherine Drummond. I'm with the Department of Chemistry, and my research is on the spontaneous evolution of hydrogen peroxide from partially oxidized porous silicon. When the silicon is in contact with water, we observe this surprising spontaneous evolution of peroxide. My project investigated the optimal conditions for oxidation and water contact time and quantified the amount of peroxide formed using a UV-Vis spectroscopy assay. Starting with re-etched porous silicon, I used both electronics-grade silicon powder with a 1% iron impurity and metallurgical-grade silicon to compare the two and look for any possible effects that the iron could have on this process. I thermally oxidized each sample at temperatures ranging from 250 degrees to 400 degrees Celsius for varying durations between 15 minutes and 4 hours. Then, the partially oxidized silicon was kept in contact with water either overnight or for 24 hours. Approximately 15 milligrams of silicon powder and half a ml of water was used for each sample. Then, to test the effects of water contact time, samples were tested in the range of immediately after making contact with the silicon to 18 hours later. All hydrogen peroxide concentrations were measured with a UV-Vis assay, using xylenol orange, which changes from yellow to purple with increasing peroxide concentration. It was determined that the optimal conditions for thermal oxidations are to anneal the porous silicon powder at 300 degrees Celsius for half an hour. The plots show the maximum concentration of peroxide at 300 degrees for a constant time of half an hour, as well as the maximum at half an hour annealing time at a constant temperature of 300 degrees. These variables can be combined into a 3D plot to show the overall optimal conditions for peroxide formation. In the future, more data can be collected on the water contact time and temperature dependence, in addition to improving the data on oxidation time and temperature. More cohesive trends will strengthen the understanding of how spontaneous hydrogen peroxide evolution occurs. The ability to produce peroxide and other reactive oxygen species is beneficial in biological applications. Silicon is a biocompatible material, and the release of silicon ions can assist with the proliferation of endothelial cells and neovascularization. Reactive oxygen species are naturally utilized as biosignaling molecules as well as antimicrobial agents to control wound infection. Combining these properties creates an effective new strategy for wound care and healing. Thank you.